So this is extensions to singular value decomposition. Okay. So hopefully you understand SVD, and now we're going to talk about some additional spectral methods that kind of build on the ideas from SVD. First of all, we'll talk about biplot. And if you understand biplot, you really understand SVD, and I think you see the beauty of this, this data transformation. So first, let me show it to you kind of what it kind of looks like, you know, in a non-biological context, but maybe is a little bit easier to do. So a biplot basically is we basically have the two the rows and columns problems, or there's sort of two ways of looking at a matrix, A or A transpose. We have them plotted in the same figure. Okay. And so, for instance, if we had an original data matrix that had, you know, each state, and it had the number of crimes or number of arrests in that state for different categories of crime, murder, assault, rape, and urban pop. Okay. We could kind of, sorry, I'm just going to stop this again, and let me just start again. Sorry, I have to always remember to turn off the timings here. Hopefully I'll remember that next. Actually, let me just do that next minute for the next one. Just let me do this right now so we don't have a headache with that. I'll just do it right now. Let's just do that. Let's see. And let's see if we can get. Okay, so these all look correct. So sorry about that. Hopefully we've figured that out. Okay, so now let's go back to biplot. Okay, so now again, we have a data matrix that has, just to get some intuition for this, we're going to start with the social science context, and we have a data matrix that contains all the 50 states and has the different crimes, murder, assault, rape, and urban pop. Okay. And now what we can, of course, do is we can correlate each state with each state, right, to make one sort of correlation, see how all the states are related to each other, or we can correlate each of the crimes over all the states with each other to see how the crimes relate. So there's two different kind of similarities we can compute, whether we're looking at rows or columns, right? And now, if we do a kind of just a PCA of one of them, or do an look at one of them, we could see all the states. And so we'd have all the states here, okay? And we'd have the different principal components. And we could see, for instance, you know, maybe North Carolina and Mississippi were sort of similar in their crime profile. And maybe, let's see, New Jersey and Hawaii are pretty similar too, right? Now we could, of course, do we could do also an eigenproblem for the different crimes. And we would see maybe that the profile over the states of murder, assault, and rape is more similar to each other than the profile for, say, urban pop. But what's really neat is we can actually project this second problem. We can take the axes of the second problem and project it back into the first problem. And so we can actually see here where all the states sit relative to these. Now are not points, they're directions. And so, for instance, we can see that, say, New Mexico has a big projection on the assault dimension, but a smaller projection onto the urban pop direction. You can see how urban pop is really almost like a different direction. So, for instance, actually, if you look here, then what you can see, which is very nice, is that across this way, you're basically looking at how states rank in terms of, let's say, violent crime. Okay, so maybe from Florida, to Idaho to New Hampshire, this is a sort of violent crime axis. And actually, up and down here on the vertical axis, this is an axis that's more dominated by the urban pop direction. So, for instance, if we go from Mississippi, North Carolina to New Jersey and Hawaii, that's kind of a degree to which we tend to get arrests for urban pop. And actually, that is, if you think about it, it's actually kind of a very intuitive way of seeing this whole data set. So now let me show you a little bit more about this and move it into a biological context. So now let's think about biological context. We have transcription factors, A, B, C, okay? And we have genomic sites. So this is our normal genomic data matrix. These could be genes, sites on the genome, whatever. And the numbers in here somehow represent the activity or binding of each of these factors onto these genes or onto these sites. Okay, so what can we do? Well, first thing what we can do is we can look at each of the factors and how does this factor correlate, say, factor A, how does it correlate with factor B? We can look at all possible correlations of this A vector and this B vector and so forth. And so we'll get this small 3x3 matrix. So then that describes how all the transcription factors are related, right? We can do another matrix. We can take all the sites in the genome and we can correlate them. So we can correlate this with this, and then we'll get a much larger, this is a 10 by 10 matrix. This is our site correlation. So that's A transpose A. And then we can do principal components on both of these things. So if we do principal components on the thing at top, now what we have is we have each of these sites and how they sit relative to the different factors. Okay, so that and so forth. And so, for instance, we have the 10 sites. And likewise, if we do principal components on the other thing, we have the 3 transcription factors, and we can ask how do they sit relative to each other. And obviously, factor A and C are sort of more similar, and there's an axis that really separates them from factor B. Okay, so we have 2 balls. But now, if you look at these things kind of carefully, look at where A and C and B and all, and it turns out, because of the beauty of SVD, we can actually just overlay these 2 graphs. And so we can take this graph and just literally biplot it onto the same thing. So here, now we have the ACB axes plotted here, or conversely, we can plot these blue points as axes on this graph, okay? So now let's look at this top graph in a little bit more detail, okay? So it tells, like I said, in one shot, you get just this beautiful understanding of your transcription factor. So let me tell you about what you could see biologically from this data decomposition. Well, okay, here's some real data. Here's now we're using real transcription factor names, not just schematized. So here we're plotting all the sites in the genome. Okay, there's many sites in the genome. Okay, and they're all plotted. And now we have all the transcription factors as axes. So for instance, POL2. Now, actually, when I say transcription factor, I'm also talking about histone modifications, H3K4ME3, TAF1, whatever, CMEC, different factors. And here's more factors, SB1, SB3, STAT1, and so forth. Now, what you can see, which is kind of, if you think about this biologically, it's very satisfying. You can see all your histone modifications and your maybe non-sequence specific things are up here in the blue cloud. You know, POL2 is not very sequence specific. These histone mods are not. And this stuff down here, STAT1, SP1, SP3, June, these are sequence specific factors. So you can see right away just the axes kind of group into these bundles where you kind of separate your sequence specific and non-specific things out. And now let's zoom in here a little bit. And so now I can zoom in, and what do you see here? Well, you can see right off that if we just think about the sort of sequence non-specific thing, we can see the sites that tend to have a big projection or tend to have big and small projections onto that axis. You know, so they really differ by the amount of sequence non-specific binding. And then here are the sites that really differ in terms of the amount of sequence-specific binding. So kind of at the same time, we've clustered all our factors, and we've also clustered all our sites in kind of a very nice principled way, and with principal components. And so I think it's very kind of cool use of this technique. And again, it's very well-established technique, this SVD and so forth. Okay, so now I'm going to go on to another extension of SVD from biplot to RCA. So RCA, it's sort of basically like doing SVD or PCA, but what we're going to do is sometimes there's a lot of noise in the data, and we want to kind of denoise the data first. So what we're going to do is, in this thing, is we're not going to directly take our data matrix and run it through SVD or run it through PCA. What we're going to do is take our data matrix and project each sort of vector in it, each column into it, onto a standard reference data set. Then we're going to make up a new matrix of the projections onto that, and then we're going to PCA that. That's RCA. And that kind of denoises things because it moves us into this kind of reference, these reference components, and we're going to find out the PCs from those first. And this is very useful for, say, single cell data. So again, in more mathematical language, we have our samples. Again, imagine we have genes, we have different conditions. And now what we're going to do is we're going to correlate these with a reference data set. So where I'm going to show you an example with, say, the GTEx data set, where we have many samples of people's RNA-seq expression, okay? And now what we're going to do is we're going to correlate these with the reference GTEx expression, okay? And so then what we have is we have a new matrix. We have each sample, okay, and how it's correlated with each of the GTEx references, okay? That's our new correlation matrix. And now we're going to run SVD or PCA on that matrix, okay? And of course, when we do that, we're going to get, as usual, sets of eigenvectors and projections onto them, and then we can, of course, look at that in some sort of PC plot. And I'll just show you. This is just I thought it's good to show you an actual data figure. So this is actually what would happen. This is taking all the data from one project that had looked at brain gene expression and just brain gene expression, and then another project for GTEx that looked at expression from all over the body and brain and projected them. And it's kind of nice what you can see here. You can see this is all the expression from all over the body, and here's the data sets from GTEx that are in the brain, and here's all the other brain. You can see that kind of nicely clusters together and so forth. Okay, so let me go on to the next extension called CCA. So I'm going to motivate this with just from the beginning with an example, because I think that's easier to kind of grasp what's going on. And I should add that the main thing with these techniques isn't really the mathematics. This mathematics is very solid. This has been developed for a long time. This isn't some new piece of math. The big thing is the intuition and really mapping it onto your problem and understanding what these transformations mean in terms of your problem. And so I think that's the thing that we're trying to do, of course, in data science application and bioinformatics, to really get understanding of specific application problems. Okay, so let's start with this problem and show how it goes. So turns out, maybe 15 years ago, Craig Venter, who's an early genome pioneer, you might remember he sort of competed to sequence the genome originally. He was obsessed with sequencing, and also he liked sailing a lot. And so what he decided to do was to take his yacht, and that's pictured over here, and sail it essentially around the world, following Captain Cook's original voyage around the world. And then what he was going to do while he was sailing around the world is he's going to drop a bucket in the ocean and get all the things, all the microorganisms, and sequence them. And this is an early metagenomics thing. And then he was going to look at how the microorganisms changed in different sites, okay? So now Now, one thing that I should say that was really neat about this thing was that now we're looking at each of these sites, we're getting the microorganisms, but also we have the GPS coordinates, we have the location of the sites. And because we have that location, not only can we look at a map, but we can get a vast amount of metadata about those sites. We can find out how deep the site is, what the temperature is, how much chlorophyll, what the salinity is, all sorts of additional things that we can find out. And that's very useful. We want to correlate that with the sequencing. So how might we do that? Well, you can imagine we're going to start off with two data matrices, as we always do. We're going to have one data matrix where we're going to get all of our environmental data. We have all the sites on the ocean. We have the temperature, the salinity, depth, whatever, make a data matrix. And then we're going to get all of our reads in each of the sites. And now from metagenomics, I won't go into detail of this. What you do here, as opposed to assembling the genomes, we kind of look at how much we're going to have of, say, either different gene families or different pathways and so forth. And so it's what's called kind of community functions. So we have a matrix now that has all the sites and basically has the amount of the reads that map onto this pathway, whatever, this particular family. So we have our two matrices, okay? Now, think about it. What we want, the biological question we want to answer is, when, say, the salinity increases, or the environment changes, how does that relate to the different microbial functions we observe? So maybe do we get more, a certain type of pathway or more of another type of pathway? So, of course, the obvious thing we can do is correlate this matrix with this matrix. So we can look for them. So we can build this huge number of correlations. And this is shown here for this particular data set. And so you can see you get some things that correlate well, but not. But it's hard. It's kind of very noisy and a very complicated picture. And of course, we'd like to simplify this in a way. So how are we going to simplify it? Well, you can't just PC these data sets, because we really have two fundamentally different matrices that we really can't put them together and PC them. So we have a slightly different thing we want to do. And I think the one thing I thought was very nice is someone motivated it with this kind of very simple analogy, and I'll walk you through the analogy here. So if we think maybe of the, we can think, let's think of health for a second and think about, we want to correlate, say, our amount of our physical fitness each week, how much we run with our weight. We're trying to lose weight. Okay, so now we have, this is like the environment versus pathways correlation. We can do a simple correlation, but really the thing we must realize is often it's not one thing being correlated with something else. It's often a whole confluence of different variables are maybe correlated together. So for instance, if we're interested in our weight, it could be how much we run, how much we cycle, how much we eat. All of those things can be correlated with our weight. And they're also correlated with many other aspects of our fitness too. They're correlated with, say, our blood pressure, cholesterol we have, and so forth. And so ideally what we'd like to do to actually do this best is we'd like to take all of this lifestyle stuff and kind of create some sort of vector or some sort of index from it, maybe a linear combination of those things, right? And then take all the fitness and create a linear combination of those things and then correlate these two things together, okay? And of course, how do we do this in an optimum way? Now, we're going to come back to our biological thing. So what we're thinking, of course, about is environmental features, somehow, looking at them all together and the different types of protein families, metabolic pathways, and so forth. So we again have these two spaces. So we have our different sites in the water, the different axes, and now we have our sites, the different families. We want to find a kind of direction in one data set that is maximally correlated with the direction in another. And that's what's solved by this technique called canonical correlation analysis. It's different from PCR SPD, which is dealing with two different matrices. And essentially what this technique does is it finds the direction in each space that maximizes the correlation, and then a secondary direction that secondarily maximizes the correlation. And then, of course, you get it's kind of like a PCA plot, but now what you do is you're plotting these two different the different variables in the space, for instance, the temperature, salinity, and so forth, how they project onto this direction. And you're also plotting, for instance, the different protein families or protein pathways, how they project onto this direction. And you kind of see these two different things together in this same plot. And obviously, things that project well onto the first canonical correlate, they'll have a very big value here and here, and likewise the second one and so forth. So it's kind of get a bullseye plot, and things that are in the middle don't have very much projection. Now, this might be all like mumbo jumbo, but again, let's think about how you could apply this and what you can get some intuition. So now this is a picture where I show you kind of the wiring diagram for metabolism. And here you can see, this is the Boehringer-Ingelheim chart. You see all the metabolic pathways. And what I've done is I've colored these pathways by their projection onto the first canonical correlate, okay? Now remember, the first canonical correlate is the thing that it most relates to the environmental variables, okay? So now what you have, it's really quite nice. You have a coloring from blue to yellow, that's the projection, and the things that are yellow are the pathways that are most correlated with the environment, and that's quite nice. So you can actually look at the chart and see which things in the sort of metabolic network are most correlated, which pathways are most correlated with the environment. And that's these yellow things here. And then just very quickly, let me show you, kind of zoom in on two of those and get some good intuition. So if we looked at photosynthetic pathways, yellow, because that's very correlated with the environment, the amount of sun and so forth. Oxidative phosphorylation, not so much. Everything pretty much has that irrespective of the environment. So a lot of intuition from this. And the key is how you take that spectral decomposition strategy, the CCA, and map it onto your problem.